Team.
Morning, everyone. I'm Frank, too, for those of you who I haven't met. I'm the Vice Chair for Quality and Division Director for the Minimally Invasive Gynecological Surgery Group, and pleased to get a chance to introduce our team from the Gynecological Research Lab. We're going to be talking today about painful periods, and excited to present a lot of work that's been done by a large team here that's grown over time here at Endeavor Health. Let me go ahead and go through the first screen.
Oh, you need to put it.
Two, you said?
Yeah, but all right. Hit play.
Screen two, you said?
Yeah, but you need it to talk. Play.
Slideshow here from the beginning?
Yeah, just
No,
there you
that.
go. Yeah.
All right. Hold on one sec. I don't think it's being shared yet. Let's see. Share screen two. Yes.
Yeah.
Okay. Let's see if share in here. And can we see the slides here now?
Perfect.
Allison?
Perfect.
Great. All right. So the title of the talk today is A Broad Approach to Understanding and Treating Dysmenorrhea. Our presenters, I'll do quick introductions for Kevin Hellman, Natalie Osborne. I've already done myself. Dr. Hellman has been the director for the Gynecological Research Laboratory. He's been here since 2010. He's a graduate of the University of Wisconsin, background in computer science. Subsequently did a PhD in neurobiology at the University of Pennsylvania. Postdoctoral studies were at the University of Chicago, Peggy Mason. Has very quickly become one of the premier investigators of visceral pain for women and pelvic disorders in the world. He's a wealth of energy and experience in doing this, and funded on a number of federal grants over the past several years. He's got a lot of data here. He's going to be showing you from some of that work that's been done here at Endeavor Health. Our colleague Natalie Osborne is a native of Canada, grew up in the Ontario area, went to the University of Guelph for undergraduate, subsequently did PhD studies at the University of Toronto with one of the premier brain imaging experts in the world, a woman named Karen Davis. And Natalie continues to have extremely strong interest in understanding how neuroimaging can be applied to the study of chronic pain disorders and underlying pain mechanisms. So two of them will be starting off. I have a little bit of work to do at the end there to introduce some new opportunities to be involved in studies with patients here at Endeavor. Real quick, in terms of financial conflicts, they're listed here. Dr. Hellman and I have some sort of fledgling support from my event for a study that we may or may not do yet. I have declarations over stock options of Maple, doing some early drug discovery, royalties for Walter Skluer for doing up-to-date book chapter, and we, as a group, have gotten research funding from Dot Lab to do some diagnostic test work for endometriosis. Natalie indicates no financial conflicts. And objective slides here, we have three components. One is to identify modern approaches to understand the modifiable mechanisms of pelvic visceral pain conditions, dysmenorrhea, endometriosis, chronic pelvic pain among those. Secondly, to critique the evidence for peripheral versus central mechanistic dominant models of dysmenorrhea, which historically has been viewed entirely as a peripheral disorder. And then lastly, to offer some thoughts on secondary treatment plans that might result in actually prevention of chronic pelvic pain in the setting of dysmenorrhea that's not easy to treat in order to optimize treatment of patients. Dr. Hellman is going to kick us off here. Kevin, take it away.
Thank you so much for the kind introduction, Dr. Tu. So first of all, I can't possibly do justice to describing the suffering of women with dysmenorrhea. It is the leading cause of missed work in school. Some people are in such severe pain they can't get out of bed several days a month. The other thing I'll point out, I'm going to use the term women throughout the talk, but regardless of gender identity, anyone with a uterus can suffer from menstrual pain. And one of the main reasons why I was drawn into pretty much exclusively focusing on dysmenorrhea is when I started to look at Dr. Tu's data. He originally designed a adaptive test to look at severity of menstrual pain, but buried within the data itself was a suggestion in this cohort of nearly 1,000 people that the worse menstrual pain a person has, the worse non-menstrual pelvic pain. So there's this very strong link in the original paper, we noticed a association reaching an odds ratio of about 13. Other people reported odds ratios of 3. But in our work, as well as others, menstrual pain is the leading risk factor for the development of chronic pain. So our hope was if we could come up with better ways of treating menstrual pain, we might be able to reduce the risk of the development of chronic pelvic pain and maybe chronic pain in general. But there was a huge problem. Our understanding of menstrual pain was severely limited. The background, I don't have much time to go get into, but you're probably familiar with the hypothesis put in most textbooks and review articles that high levels of prostaglandin during menses, as well as oxytocin, lead to increased uterine contractility, which causes ischemia, and which in turn causes pain. Hopefully, in the next 10 minutes, you will realize that this conventional hypothesis is oversimplified, and there are many uterine as well as non-uterine mechanisms. I hope by the end of this talk, you'll have a whole body understanding what are the mechanisms responsible for menstrual pain, and it's not just about the uterus. So what we did, which was substantially different than anyone else before us, is we decided to look to see what's going on during a cramp using non-invasive methods. We've published a lot of the data on smaller data sets. Right now, this contains our data set all the way up through the summer before last. It takes a while to analyze this data. We have nearly 100 primary dysmenorrhea participants and about 30 healthy pain-free controls in this cohort. So I'm going to show you what occurs during a uterine contraction. So I'd like you to note the pseudo-color T2 weighted image on the right, as well as the original T2 image on the left. And what you're looking at here is an MRI captured every 2 seconds, speeded up by a factor of 4. And what you're going to see is a thickening of the myometrial layer that occurs. They last about 40 to 70 seconds. It occurs 3 to 5 times every 10 minutes in people with menstrual pain. So these are visible on MRI. We only see these during menses. And before this, it really was not known what occurs when a woman is experiencing a cramp, except when they try to use something like an intrauterine pressure device. So an easier way of looking at is looking at a cross-section of this MRI over a 10-minute period. And what you see here are symmetrical. It's both anterior and posterior myometrium. You see these decreases in signal intensity, and what you see beneath is the participant squeezing a pressure bulb. So you can see, at least this first event, there's a lot of pain at the beginning of this contraction. Here it is somewhat delayed, and here you don't even see anything at all. So there's not necessarily a one-to-one relationship. It previously been believed that women with dysmenorrhea have high pressure uterine contractions and pain-free controls do not. But in fact, the vast majority, more than 50% of our pain-free controls do have uterine contractions. Admittedly, nearly 90% of our primary dysmenorrhea participants have a uterine contraction in the 10-minute window we observe with MRI. We also observe these events in people with endometriosis and fibroids, and I'll point out we see absolutely nothing in the fibroid per se in terms of changes in signal intensity. It's exclusive to the myometrium as well as the endometrium. So what we do see is the contractions are definitely more frequent in our participants with menstrual pain as well as endometriosis and fibroids. There's something about increased uterine contractility that could be causal, but it's not mutually exclusive because certainly our pain-free controls have no pain, but they do have uterine contractions. So when do these painful events occur? So in some of these people, in some of these events, this is what an average contraction looks like. You see pain. This is when they're squeezing the bulb, the percentage of participants in this particular phenotype. Some of them squeeze the bulb at the very beginning of the contraction, and some of them we see a delayed phenotype that could be due to reperfusion or ischemia. And sometimes we see in people both kinds of events. We're still looking at our full cohort to figure out how many people fit into each of these subtypes. So what could be responsible? Recently, I mean, in the last 3 or 4 years, the Nobel Prize was awarded for the discovery of piezo, which is a mechanoreceptor. It's located not only on the myometrium, but also the nerve cells that innervate the uterus, as well as the vasculature. So it's widely believed that this mechanoreceptor could be responsible for menstrual pain. Recent data that was shown at the International Association for Study of Pain showed that if you make mice without these piezomechanoreceptors, they'll have different issues with parturition, and the animals have insufficient contractility, and not all of the pups will be born. So there's something very critical about the role of these mechanoreceptors in parturition, and they likely play an important role in menstrual pain. They're typically coupled or associated with TRPV. You might be familiar with TRPV because it's responsible for the sensation of capsaicin or hot sauce. And this is also located on C fibers, and TRPV expression is correlated with severity of dysmenorrhea, TRPV levels in the endometrium as well as the myometrium. So this could be one of the causal mechanisms responsible for menstrual pain. It might be a combination of increased uterine contractility as well as the presence of these mechanosensitive channels. The leading hypothesis is that there is a role for ischemia in menstrual pain. The thought is maybe sustained high pressure events or the breakdown of the endometrium leads to profound ischemia. To investigate this, we used two different methods. We used a method known as R2 star imaging with MRI, which allows us to look at the relative amount of perfusion and oxygenation. And what you're looking at here are R2 star images of a participant with dysmenorrhea as well as a pain-free control. And what you're looking at here is Doppler ultrasonography of the uterine artery. And so we did that while participants were on their period before and after a therapeutic dose of naproxen. Enormous amount of data to go through. So instead of showing all the box and whisker plots, I'm just going to show you a summary figure. So what you'll notice here, what indicates in red is the relative level of perfusion and oxygenation. And you can see in people on their menses have a reduction in perfusion and oxygenation, but our participants with menstrual pain, if anything, have more perfusion and oxygenation than our pain-free controls. They also have higher uterine arterial flow. So it's not a matter of impaired uterine arterial flow. Not only that, when we give them naproxen, which is ordinarily therapeutic, naproxen is an NSAID, we see even less perfusion oxygenation, and our participants, our pain-free controls, have the exact opposite response. So what we believe and what we published is that it's likely to be uterine inflammation rather than ischemia that's contributing to menstrual pain. Now, admittedly, these R2 star images are sustained over 30 seconds to a minute. So what you really want to look at are transient events, but certainly menstrual pain is not due to sustained ischemia. It still could be due to transient ischemia, but certainly not as a state. And if anything, there's more blood flow suggestive of an inflammatory event. One of the ways to look at and see what occurs transiently is to look at the Doppler arterial flow during a cramp. And we see no changes in the pulsatility index or resist index during a cramp. So menstrual pain is not due to an impairment in uterine perfusion. Another way of looking at these transient events is to look to see what happens to a uterine oxygenation continuously. So one of the ways of looking at uterine oxygenation over time is to use BOLD imaging, blood oxygen level dependent imaging. And just as a demonstration, because I believe we were the first to ever do this, we gave participants supplementary oxygenation for 2 minutes. And what you can see is when we give them supplemental O2, there's an increase in their uterine oxygenation or their BOLD signal. So we learn a couple of things from this. First of all, the uterus is not sitting at 100%. So it's not, there is some desaturation of the uterine oxygenation. And two, unfortunately, we asked the participants whether it helped their menstrual pain, and it did not. So just supplemental oxygenation alone is not enough to help their menstrual pain. So this is just examples from a couple of participants. And what you can see here in red is, oh, is my mouse not showing you, right? You see these transient dips in uterine oxygenation on the right. And what happens is we believe these events are responsible or linked to some of the cramping pain. And after naproxen, you can see it goes away. We do not see these transient dips in uterine oxygenation and pain-free controls. So it might be that transient ischemic events are responsible for some of the menstrual pain when we use bold imaging. It's not just about the uterus. Initially, we had intended to record cervical electrical activity, but we found really strong abdominal signals in our participants. And interestingly enough, you can see this is an abdominal EMG, and this is their spontaneous report of pain. This abdominal EMG event precedes their report of pain by 1 or 2 seconds. So we believe, based on what we saw in some of the data in some of the people with simultaneous uterine ultrasonography, is what was known in animals. What occurs is this, is right after a uterine contraction, you get a visceral motor reflex or a guarding reflex, which leads to intense abdominal muscle activity. And if you can imagine, it's like doing a sit-up every couple of minutes. And just the abdominal muscle activity alone might be one of the causes for menstrual pain, which might explain why hot water pads or heating or transcutaneous electrical nerve stimulation to the abdominal surface musculature might help with menstrual pain. More work needs to be done on this. So another thing to think about is it's not just about the uterus. Dr. Tu developed a method of looking at visceral sensitivity non-invasively by just having people naturally drink water. And what we found in her earlier work, as well as her follow-up work, is people with menstrual pain are more have lower bladder tolerances than pain-free controls by roughly 100 milliliters. So they have less visceral tolerance, but they have quite a bit more visceral pain. So these participants with menstrual pain, they have at all these standard systometric levels with urine in their bladder, more pain than a pain-free control. So I know it's a little bit backwards to talk about this now, but certainly there's something going on in the uterus. So the original hypothesis was that elevated levels of prostaglandin are responsible for increased uterine contractility. So one of the problems with the prior literature is they typically look at people with older adults that might have secondary dysmenorrhea. If you look at adolescents, so it's very unlikely within the first year or two that they necessarily have endometriosis, or very unlikely that they have fibroids. We can look at prostaglandin levels in their effluent. So this is data from over 50 people. It's one of the largest studies ever conducted. And you can see that we find higher levels of prostaglandin F2 alpha in their effluent, but certainly it's not enough to explain. You can see by the confidence intervals, it might be significant. They might have roughly 2 times more prostaglandin in an effluent, but it cannot perfectly predict who has menstrual pain and who does not. So prostaglandin alone does not explain why some people have menstrual pain and others don't, but certainly it could be a contributing factor and it is elevated. We've begun to do some work looking at other molecules, and we've identified other eicosanoids, leukotrienes. There are certainly other molecules that are expressed at higher levels, as well as changes in some other metabolic pathways that would suggest there's reductions and breakdowns of some of these products that could lead to menstrual pain. So in short, prostaglandins likely contribute to menstrual pain, but there's likely many other molecules, and you would need other molecules because prostaglandins alone cannot explain why some people respond to NSAIDs and other people don't. So what's another reason why some people might respond to NSAIDs and other people don't? Well, we've looked to see how much of naproxen is absorbed, both in people with primary dysmenorrhea, shown as DIS, as well as second dysmenorrhea, DIS-S. And what you see is in the participants with primary dysmenorrhea that have poor analgesic responses, they absorb very little of naproxen. So one possibility is they're hypermetabolizing naproxen, but we find very little levels of their metabolite. So this would seem to suggest that people with dysmenorrhea that do not respond to naproxen, whether they have primary dysmenorrhea or secondary dysmenorrhea, they're not absorbing the naproxen. Another important inference from this data is the more amount of naproxen in their bloodstream, the better analgesic and effectiveness they'll get. And as we'll discuss later on, there is some consideration about dosage. So in summary, I've shown that it's not just about prostaglandin. There's likely other molecules that are responsible for menstrual pain. Instead of causing ischemia, it likely causes uterine inflammation. We found a lot of ischemia in our pain-free controls work in heavy menstrual bleeding by Critchley has shown that uterine ischemia can be protective. So we believe something very similar is going on in our pain-free controls in this study, and it might be that ischemia is actually a protective response. Certainly, uterine contractions could be responsible for pain, but it's not the sole mediator because our pain-free controls have contractions but don't have pain. So you need a combination of contractions as well as some mechanism for visceral sensitivity, which might involve the TRPV or piezomechanoreceptor. Also, it's important to note we are seeing evidence of transient ischemia as well as some changes in the abdominal musculature. So it's not just about the uterus. There are some widespread changes in other organs, and we will continue to explore the role of the whole body effect or whole body mechanisms in menstrual pain in the next section.
Good morning, everyone. Okay, so I'm going to talk to you briefly about central mechanisms of pain and dysmenorrhea. So I'll talk about some of the behavioral pain measures that indicate there might be altered central processing of pain and dysmenorrhea, tell you about what neuroimaging reveals with respect to abnormal brain structure and function in this patient group, and talk about whether combining brain and behavior measures might be able to identify patient subtypes and predict future chronic pain risk. So ultimately, the goal is to be able to identify the unique combo of peripheral and central mechanisms that are contributing to pain and dysmenorrhea with the hope of tailoring better treatments. So using the bladder filling task, created by Dr. Tu, we identified a subset of women who have dysmenorrhea and bladder pain. So you can see here their pain ratings at various stages of bladder filling, and the provoked bladder pain in this subgroup, which we call Dys-B, was as bad as that reported by people who have diagnosed bladder pain syndrome. And something else about this subset of dysmenorrhea patients is that they have worse pelvic pain in general. So pain with urination, with bowel movements, with intercourse. Another study in our lab found that women who have dysmenorrhea and this provoked bladder pain were more likely to have IBS, and that the presence of bladder pain was the strongest predictor of new onset IBS at 1 year. Now, if we do pain testing at the primary site of pain in dysmenorrhea, the pelvis, unsurprisingly, we do see lower pelvic pressure pain thresholds in people with dysmenorrhea. So you can see in the graph is the amount of pressure required to elicit pain, and it is lower in dysmenorrhea, and the same is true for people with Dys-B and people with bladder pain syndrome. And at this primary pain site, the pelvis, we also see that people with dysmenorrhea and dysb also report more pain with tampon insertion. So I'm going to tell you about another tool that we can use to measure central pain processing, and that's a task called conditioned pain modulation, or CPM. There are a lot of variations of how to do this in the lab, but every CPM will involve a test stimulus and a conditioning stimulus, which is usually a different pain modality. If you could imagine you're sitting in your chair and I apply a painful pressure to your right shoulder. Then I ask you to stick your left hand in a bowl of hot water. And while it's in that hot water, I again apply that painful pressure to your right shoulder. How many of you would guess that the painful pressure would seem worse when you've got your hand in the hot water, or would it seem better? Well, what we actually find is a pain inhibits pain phenomenon in some people. So when they rate the pain of the test stimulus initially, it might be a 5 out of 10. But when we ask them to rate that test stimulus again, whilst they're having the conditioning stimulus, aka their hand in the hot water bowl, they'll actually rate the pain a lot lower. This CPM effect is thought to reflect engagement of our brain's descending pain modulation system. And what the field has noticed is that this response, it tends to be impaired in some chronic pain conditions. CPM can predict future pain, for example, who will develop chronic post-surgical pain, and it can even predict treatment response to certain drugs. So when we apply these experimental pain measures in our subset of dysb patients, we actually see widespread pain sensitivity. So not just at the primary pain site of the pelvis, but actually we found lower pressure pain thresholds at multiple body sites, forehead, shoulder, hip, and knee, in this subset of dysb. And when we performed CPM, we found reduced or less efficient CPM in this subgroup, suggesting they are less efficient at engaging their descending pain inhibition. So that leads us to the question, what could be causing this widespread pain sensitivity and this impaired descending pain modulation? Perhaps there's something different about the brains of people with dysmenorrhea and dysb. So there have been studies that look at structural brain differences in dysmenorrhea. So here's a graph, and in red, you can see areas where the cortex was thicker in people with dysmenorrhea compared to healthy controls, and in the blue, areas where the cortex was thinner. And some of these areas are classically associated with pain, like the insula, the anterior cingulate cortex, the medial prefrontal cortex, the secondary somatosensory area. So that reflects some of the things that we see in the wider chronic pain literature of structural brain differences. And what's interesting is some of these differences were also related to the behavior, so the actual pain experience of the people with dysmenorrhea and how long they'd had pain. And that's interesting because when we do these cross-sectional studies, it can be a bit of a chicken and egg scenario. So we don't know if these brain differences were always there and led to the development of pain, or if the brain differences came about through maladaptive neuroplasticity after having pain for a certain amount of time. Now, what about functional brain differences? So this is a study that looked specifically at the periaqueductal gray, which is a really key region when it comes to ascending and descending pain processing. And they looked at something called functional connectivity, which is a measure of how correlated activity in one brain region is to another. And we use this functional connectivity when a person is at rest to get a sense of the functional architecture of their brain. How are their various brain regions communicating with one another? And here in the dysmenorrhea patients, they saw widespread differences in how this key brain region communicates with various brain networks. So the default mode network, frontal brain regions, somatosensory brain regions. Again, it's a chicken and egg, so we don't know if these differences were there before or after developing dysmenorrhea. So something else that we can do when it comes to looking at brain function is look at evoked activity. So how does the brain actually respond to a painful stimulus? So this is a small study out of a group at Oxford that found altered central processing of somatic pain. So they applied a thermal stimulus to the lower abdomen of people with dysmenorrhea and healthy controls, as well as another site on the forearm. And on the left are the brain activations in the control women and the women with dysmenorrhea. And what they found is that there was less deactivations in response to the somatic pain in the women with dysmenorrhea. So they weren't deactivating the same brain regions as the healthy controls. They also saw widespread increased sensitivity to heat pain at both the lower abdomen and the forearm in the dysmenorrhea patients. Interestingly, they measured cortisol and they found decreased cortisol in dysmenorrhea, and the level of decrease correlated with how long they'd been in pain, leading them to think that there might be altered hypothalamic pituitary adrenal axis function in dysmenorrhea, and that's something that we see in chronic pain patients. They also found altered pain-related brain activity in dysmenorrhea, and that was seen across the menstrual cycle. So it wasn't just when they were menstruating, but also when they weren't menstruating, specifically in the entorhinal cortex, which is interesting because that area, as well as the hippocampus, is gaining new attention for its role in pain chronification. But basically, the fact that these changes were seen across the month indicate that there might be state related and trait related changes in brain activity in dysmenorrhea. But what about visceral pain? So the evidence for visceral pain differences is a bit more mixed. So there was one study that did find that patients with dysmenorrhea are more sensitive to painful distension of the sigmoid colon and rectum compared to healthy control. Whereas another study found no differences in pain ratings or brain activity during rectal distension between dysmenorrhea and healthy controls. But something that we're interested in is what is happening in the brain and behavior of different subtypes of dysmenorrhea. Are there differences in central processing between DIS and the subtype DIS-B? So this is part of a larger study we'll be doing and is ongoing, where we're recruiting people with dysmenorrhea, people who have DIS-B, and healthy control participants, and then following them up a year later. So we've already seen in our older data behavioral signs that there's altered central brain processing in DIS-B with that CPM task. So we'll be doing the CPM task again. So we'll be having a pressure cuff on one of our participants' legs and have their foot experiencing very cold water pain. And we'll see if we again see that impaired CPM response in our DIS-B. But the twist is, this time we're going to do it in the fMRI. So we can collect at the same time their brain activity in response to this experimental pain test. So if we're imaging these people, we can collect information about their brain structure. We can collect information about the functional organization of their brain at rest and information about their brain's response to pain, and start to answer questions like, do people with DIS-B show abnormal brain responses during the conditioned pain modulation task? And do brain abnormalities predict future chronic pelvic pain? And with that, I'm going to pass it over to Dr. Chu and talk about more future direction.
Thank you, Natalie. Thanks. Yeah. Well, hard act to follow when you have colleagues who are brilliant scientists like Dr. Osborne and Dr. Hellman. But one of the fun things about this role here is I do get a chance to talk about translation of these findings into clinical applications, and also to talk about where the lab's going with some new and exciting work. So, given the backdrop of the extensive work that's been done here to look at both peripheral and central components that might contribute to chronic pelvic pain vulnerability through the lens of dysmenorrhea, one thing that Dr. Hellman has been very excited about and been very intentional about is trying to actually see if we can do potential secondary prevention studies for chronic pelvic pain. And in two sets of pilot studies right here that are here logged in clinical trials, our group actually looked at trying to reduce menstrual pain and to see if we could in turn reduce some of these potential correlates or biomarkers for chronic pain, analogous to say reducing cholesterol levels in a patient ahead of them actually developing a heart attack. And this is a phenomenon that, in terms of thinking about doing chronic pain prevention, it has not really been something we've attempted in women's health yet. There have been attempts to try to do this in post-surgical pain, but we're really far behind there. These studies are combined in this graph here, and actually, what's very encouraging about this is in using the bladder pain assay that we described that was developed some years ago here, we were able to use that essentially as sort of a proxy for potential central sensitization, or at the very least, sort of visceral convergence of pain, some sort of extent of spread of pain risk, and show that if you do more optimal management of young women with moderate to severe dysmenorrhea, that after 6 months of treatment, whether it's using effective doses of naproxen, this is typically 500 milligrams twice a day, or maybe 550 twice a day, or if you give a patient birth control pills, whether you use it in a continuous or cyclical fashion. In both these small pilot studies, average maybe about 15 to 20 patients maximum, there was a significant reduction in the abnormal bladder pain sensitivity in patients who have DYSB. And this is really one of the first indications eventually we might be able to actually reverse the trajectory of pain vulnerability in individuals. Limitations, of course, these are very small samples in a pilot study. And even if this does prove to work, there are a lot of patients who will not respond to naproxen or can say that they can't tolerate birth control pills or don't get a response. So this is definitely something that we're going to be working on more. In parallel, the patient-reported outcomes that one might see tend to be looking in an adverse fashion amongst chronic pain patients. In many of these cases, following the use of naproxen, in this particular example, also improved. You can see that not only did menstrual pain improve markedly from a moderate level down to more of like a mild level, from a 7 to a 4. Non-menstrual pelvic pain scores, these are low because these are non-chronic pain patients, also improved after 6 months. And sort of all the other similar things, pain impacts, overall symptom profiles, and even like body map total pain scores all were significantly lower after something as simple as making sure that you give adequate treatment of painful period. Now, these are relatively modest, but you can imagine this effect might be amplified or magnified if we were to do this systematically and to start early on when a person first recognizes they're having significant menstrual pain distress. And to our knowledge, there's by no means no systematic efforts to do this in the United States. When a young woman chooses to start to treat her menstrual pain effectively is very much sort of a grab bag, and something that I think Dr. Hellman is particularly interested in trying to change. One thought that he has in a trial that we're going to be presenting here that may be effective is to see if we can do some of the prediction of the onset of menses and try to time the onset of effective treatment from the get-go. And there are a number of these devices available on the market now where look at urinary assays, may be able to identify metabolites such as pregnane, diol-3 glucuronate, the byproduct of progesterone metabolism, that a substantial number of women will have a drop in those levels if you measure them on a daily basis at the immediate onset of menses. And so instead of a woman having to take high-dose NSAIDs kind of around the clock, they could kind of look at when the urinary marker transition occurs. A relatively straightforward study. Dr. Hellman is going to be looking at that in an upcoming study, which is really exciting. There are probably some other markers we can pair up with that and make this even more precise for prediction. And so there are a number of exciting opportunities that if you're thinking about a prevention of chronic pelvic pain lens and optimization of painful period treatment lens, they are both independently worthwhile goals. Doing them together means that you probably can get a really effective reduction in chronic pelvic pain if you can optimize menstrual pain treatment in many women. Dr. Hellman spent a lot of time in his study about looking at all these potential physiological targets. And I think his work has been incredible, but he's also emphasized that some of this stuff really has to be done much further before we can talk about specific treatments. For example, we can't put everyone on high-dose oxygen, and there have to be, a lot more advances in some of these innovations. But this pilot work with naproxen, the use of simply full-strength naproxen at prescription levels every 12 hours appears to be quite efficacious. Timing to the ahead of the onset of periods could be even more exciting. Dr. Hellman's pointed out that even as a scientist, he recognizes the practical challenges of actually getting this work done clinically. And patients will probably do better if you just give them a single pill instead of have them take two over-the-counter pills. Some practical solutions here. There may be a subset of patients that, akin to how we do things in depression, where doing looks at metabolism of these medications may be important too. Some of these patients, as Kevin's earlier slide showed, may need to have their naproxen concentrations measured over time to understand if they actually would benefit from the right dose of naproxen because they're hypermetabolizing it. I do like this strategy about potentially looking at urinary assessments in an ambulatory setting to figure out the right time to pre-medicate. And I think a lot of patients will be very receptive to this. There are a lot more other things that we could do that are beyond this talk. Work by Gian Bernardino and other colleagues of ours have indicated that treating one pain condition that's linked to another pain condition can work in either direction. So if a patient has irritable bowel syndrome and has menstrual pain symptoms, you might want to, say, do dietary interventions or do more mindfulness work to work on the IBS that might in turn reduce the organ impact onto the uterus in turn. And the collective work might reduce a person's overall chronic pain vulnerability. There are obviously a lot more molecular targets. Dr. Hillman has promised me that a couple hours now we're going to be looking at some new exciting data looking at menstrual effluent that's been collected from the pediatric study, but I don't know what it's going to say yet, so I can't tell you what he's got up his sleeve, but it sounds very promising. And with that, the sum total of all the work that the Gynecology Research Lab, a lot of people think at the end of this, has resulted in some additional grants and new studies that are starting. We'll start first with the renewal of the original CRAMP study, which is now CRAMP 2, where we identified this chronic pain vulnerability phenotype. We call it DYSB, but again, it's this idea that you have both significant menstrual pain and you have bladder pain sensitivity, but you don't have chronic pain. And in that trial that started about a year ago, and that's been interrogated very aggressively by our amazing research coordinators, Dina and Sarah, we have 3 different aims. We're going to look at, in fact, this novel phenotype of vulnerability, this dysmenorrhea bladder pain, and say, what happens to this over time? Some people will have it and it will probably go away. Some people will not. And can we identify who those people are and why they go that direction? Secondly, we recognize that in not being able to just identify the full-blown effect of chronic pain, that we can look at these surrogates like DYSB or bladder sensitivity. And we can also look at your overall relative sensory sensitivity, a condition we call multimodal hypersensitivity. This is done typically in a laboratory setting where we can control the external stimuli. And we're going to try to look and see, using some of these measures like quantitative sensory testing and EEG, to understand what results in a person having more bladder sensitivity or more overall sensory sensitivity. And unfortunately, we don't have as much time to talk about this, but an exciting addition to the team has been to potentially look at the impact of TLR4 mediated inflammation and how differences in one person's relative ability to mount an inflammatory response versus another might be also at the heart of having chronic pain vulnerability. I recall that Dr. Hirsch's lab has worked on this in the past in relation to looking at preterm vulnerability or preterm labor vulnerability. And these TLR4 pathways are a really exciting place to try to look at additional causes that maybe would underlie, stress or latent infections that we don't understand that could also explain why some of these patients are at high risk for pain. There are 2 aims here in particular, and I just want to really show that we're going to be trying to get almost close to 1,000 people here. If you guys in general practice happen to have patients that have painful periods and like to participate in the study, we have a pretty large-scale advertising strategy across the city, but we're always happy to take our own home patients as well. We're relatively early into the study at the year plus, but the teams are ramping up markedly the recruitment of patients. You can see that we have a pretty aggressive target in the spring, but we're up to almost 100. And importantly, we're finding the vulnerable group, which is not easy to find out of thin air since they don't know they have vulnerability. It'd be like trying to find someone with hypercholesterolemia who doesn't get a blood draw. And there'll be a lot of opportunities to look at other important covariates and the space constructs, all of the factors that oftentimes are related to chronic pain, emotion, cognition, fatigue, etc. There'll be a lot of questions they're going to fill out over the next couple of years. The second parallel study to this study, which one of the cool things about the first aim is it's being done entirely in the ambulatory setting. The patients will do their bladder assessments at home, but a subset of them, as you can see, about 200 will be coming into the lab to be doing the more intensive quantitative sensory testing that Natalie showed you, as well as a parallel to fMRI using high-density EEG assessments. And we believe there'll be a chance here. You can see there's already about a good almost 10 to 15% of those patients that have been initially enrolled and brought into the lab. Then we'll get a chance to look at a lot more of these actual underlying mechanisms at a much more precise level than simply just using the PROs. Probably even more exciting in my mind is that the government has tried to solve the opioid pandemic by spending a lot more money on additional studies to look at the underlying etiology of chronic pain. The HEAL initiative is something that hopefully all of you are aware of. And Dr. Hellman, through some incredible work, has recently gotten an R01 to do a follow-up study of some of his pilot work on NSAIDs and how NSAIDs might be able to be harnessed as an effective tool to reduce chronic pain risk. And in his NSAID HEAL study here, he's going to be doing a parallel study to what we're doing with the mechanisms of the DYSP phenotype, and actually look and see if we can do an extensive predictive model of how does a person have worsening of their overall pelvic pain symptoms, essentially as like almost like a symptom precursor to development of full chronic pelvic pain. And one thing that's really exciting about this study is that Dr. Hellman is using a person's actual response to a year of optimized use of naproxen at the dose we talked about earlier to see if that, in relation to other factors such as some of the sort of the sensory tests we talked about or some of the underlying mood correlates, would predict the direction that your overall pelvic pain symptomatology goes before you actually have full-blown chronic pain. And it's really a neat opportunity because people have really never tried to look at NSAID responses that predict. There are two aims there, and he has just started doing the actual IRB work for this, but this is going to be another exciting study we're going to be recruiting for. I think it's a good point for stop here. We need to thank our colleagues here. There's a huge list here. We have some senior investigators like Dr. Gebhardt, who's an elite scientist that hangs out with us in his retirement. Dr. Prasad in the neuroimaging group, as well as Dr. Ken Holly, who has recently joined us from Dr. Hirsch's lab. Enormous group of research coordinators, former students, former residents, but particularly want to highlight the work of Ellen Garrison, Sarah Darnell, Tina Bavritsis, and Katie Gervais, who are our current in-house lab group. Just been unbelievably passionate and efficient in getting some of these critical studies done. A lot of other collaborators, other sites here that we can't go through all the work in thanking, but primary funding has been through NICHD as well as the NIDDK through these grants here. And I think Dr. Hellman, Dr. Osborne want to take some questions here with the remaining time we have. So thanks for all your time. I think you can use the function if you have any. So we probably got a couple minutes here to talk. Yeah, we've got a few minutes. We're finishing a little bit ahead of time. So let me pull this up here. Kevin and Natalie, you probably have lots of questions of your own that you'll probably ask each other, but let's see if we got anything in the chat here. Just real quick, there is a link for the Survey Monkey and also the information to put in for your CME credit. So thanks for your attention. Any questions from the audience? I will actually I'll swap out seats here and let Dr. Cochran sit in the hot seat here, but I have a couple of questions for clarification since we're on here. But yeah, Howard, come on over. Kevin, I think one of the questions I have is one of the opportunities to work with a really elite scientist is like the work that we've done here over the last almost 14 years. I think actually it has been 14 years since we were in Montreal together. What are the most important studies that we need to replicate from what's actually already been done to this point in your mind to really, cement these and get people to kind of take some of this stuff much more seriously?
So there's several. So first of all, there are studies that have shown that people with dysmenorrhea, when they take naproxen, there's a reduction in their when they take naproxen, there's a reduction in the prostaglandin in their effluent. It's a good question whether that predicts response to NSAIDs. The naproxen not only needs to be absorbed, but theoretically, it's acting on the uterus, and you'd expect a change in the prostaglandin levels in the uterus. There's another possibility that naproxen might be acting centrally. Certainly, it penetrates the brain. It crosses the blood-brain barrier. People should know that a lot more work has been done in neuroscience about changes in the brain during menses. It's kind of funny. If you go to a neuroscience conference, you'll see all the different molecular signaling pathways in the brain. that cause increased prostaglandin levels in the brain. And interestingly enough, it has an important implication. So it's not just animal models, but people as well. If unfortunately a woman on her menses is in a car accident and has a concussion, she's more likely to have a protective effect. So prostaglandin might actually be having a protective effect on the brain. So it's a good question, what's going on in the uterus? We know much more about how estrogen and progesterone affect uh, expression in the brain of prostaglandin as well as other inflammatory molecules. It's a good question. Why hasn't such work been done on the uterus? Some people blame a lack of funding, but I would say there's also a lack of other scientists that are explicitly studying what's going on in the uterus during menstrual pain. Part of the problem is that when Vioxx basically got sued out of existence, all the world's best experts on inflammation also disappeared into retirement. So we need renewed interest in inflammation, and I think there is some widespread interest in inflammation. And if more people are willing to study uterine inflammation, that would be great. There's some great work at neuroscience that's really linking inflammation to pain. So I would say our current work studying linking inflammation, just looking at what's in the effluent as well as systemically, there's been limited work looking at systemic markers of inflammation. I didn't have time to share it today, but Dr. Kayantali has found that some of the same inflammatory markers, heat shock proteins, that have been found to be altered in people that develop preterm labor are also altered in people that have menstrual pain. There's likely a shared mechanism between menstrual pain and preterm labor involving inflammation.
Wonderful. I don't see any other questions in the chat box. I will throw out one other little interesting conversation piece that Dr. Hellman and Dr. Osborne had. Kevin was asking us about, are there other treatments that maybe are, on the horizon that might, really make a dent in this? And I will say the caveat being that we've just shown you that it could be quite complicated how a person experiences menstrual pain, not just the simple prostaglandin story anymore, not just simple contractility. And then likewise, the chronic pain we all know is relatively complicated, involving both periphery and the central components. But a recent study that was published in 2021 by a French group that's kind of been known as being sort of a pelvic neuroscience group, they did an observational study of women who they had already worked up for any other explanation for their menstrual pain, normal ultrasound, normal laparoscopy, very severe dysmenorrhea, significant dysphoria. They ascribed a very intense amount of menstrual contractions. They actually cited your studies earlier on, Kevin, in describing this phenotype. Then they call it painful uterine syndrome. When they took a hysteroscope and injected 100 units of botulinum toxin into the front wall of the uterus and 100 units into the back wall of the uterus, these women, the N of 30 study, they got like astonishing reductions in pain scores in this very carefully phenotyped group. They had like their menstrual pain went from an 8 to a 2 after a couple cycles, and their pain with intercourse, even more interestingly, went from a relatively severe level to a relatively mild level. Do you have a sense of how many people that are in our studies would actually meet that sort of a phenotype? Is this truly a bit of a unicorn, a person that truly has no other cause for their period pain? You think there's a lot of people out there that might benefit from doing botulinum toxin treatments?
So based on what you said, so I have a couple of thoughts about that. So, the interesting question is what percentage of people would respond and how long the effect lasts. The important thing to note, which we didn't talk about here, is there is an enormous placebo effect when it comes to treating menstrual pain. And it may not be entirely, did they do a sham control?
Of course not. No.
Or is that, yeah. So
but an 8 to a 2 is not a casual amount of pain reduction.
So the funny thing is to some extent, you do get these really large placebo effects that make it hard to do good research. But what might be responsible to some extent for the reduction in pain with sex, we certainly see in our own participants with primary dysmenorrhea increased pelvic sensitivity. And it's known when a woman has an orgasm, or it's believed when a woman has an orgasm or is having sex, there's changes in uterine contractility. We didn't have much time to go into it. There's two kinds of contractions. You have peristalsis and these sustained uterine events. They're often mixed up in the literature, but it's believed that these contractions as well as peristalsis plays an important role in fertility. Definitely been understudied. So it could be that these Botox injections are affecting not just uterine contractility on their period, but peristalsis throughout the cycle.
Mm.
So one of the important concerns, obviously, with that Botox study would be long-term toxicity of the reproductive system. And I think a lot of people would be hesitant to conduct such a study, but if it works, that would be fantastic because honestly, Botox has a pretty good safety profile.
That is a good time to just remind people that that is certainly an off-label use of botulinum toxin, and not to be suggested that the FDA has approved that. I don't see anything else in the chat box, but I appreciate you guys all coming to this talk. I want to really thank the work from the Gynecological Research Lab. That list of collaborators is enormous. We've had unbelievably generous people, contributing their time and energy and intellect to get this stuff done. And Dr. Hellman has, I think, been very, very richly rewarded for deciding to come stick around with us for a few years, and he certainly made quite a run of it. And looking forward to seeing a lot more from Dr. Osborne's contributions. Thinking we'd have neuroscientists talking about women's health when I first got here in 2004. 20 years later, it's I tell you, it's a little bit pleasantly surprising. I was an undergraduate, and I just assumed that was a chapter of my life I'd close out when I left Vanderbilt in 1993. So there'll be a lot more to see here. Well, we appreciate your time, and we'll give you guys a minute back here. Allison, thanks for setting us up.
Richard has put something in the chat that I would like to at least read because it's very
oh, please do.
Yeah, congratulatory. He says his audio isn't working well, but he just wants to say that it's phenomenal work on pushing forward our understanding of pain mechanisms. I think most importantly for patients, this really speaks to their own lived experience with how peripheral and central nerve factors contribute to their pain as well as response to treatment success. So really important work on so many levels and so many subspecialties of our OBGYN department. So thank you.
That's very kind of Richard. He is the author of one of those papers, and I think he's probably too modest to recognize that's his own work that we're citing. But no, hopefully a lot of hope here and a lot of promise. And Kevin says he wants to eliminate pelvic pain probably by the end of the decade. I'm guessing he probably wants to do it by even sooner. But I think there's definitely some opportunities here and some real neat approach. So thanks for that again. And yes, thank you, Allison, for passing that along. All right, everyone, have a great rest of the day and happy Halloween.
Thank you. Take care now.
And make a few comments.